Hello. I'm glad to be here, and I'm going to tell you about Ragout, which is a tool for reference-wisted assembly. And our key feature is that we can use multiple references simultaneously. But first, I want to start from a short story. In Russia, we have the longest railroad in the world called Trans-Siberian Railway, and it is more than 9,000 kilometers long. And it has 15 million railroad ties in it. And one day, aliens have been landed near this railroad. And you know, aliens are big jokers, so they have painted the ties in different colors. And after, they took a lot of pictures of their work. And then, aliens had been gone, and rain fell, and it vanished all the ties from the railroad. The question now is, can we reconstruct the coloring by using these pictures Alien gave to us? Probably all of us here know that we can apply Genome Assembly here, and number of ties is somehow comparable to number of nucleotides, so we can apply an assembler for bacterial genomes, like spades. I will start from assembler. What do they do? Is actually they join small overlapping DNA fragments called reads into chromosomes. But unfortunately, in most cases, assemblers fail to reconstruct the complete sequence. Instead, they output contiguous regions of chromosome called Contigs. They do not overlap. They cover the entire genome, but we don't know the permutation. For some studies, it is already enough, but other requires complete sequence. What do people do to get a complete sequence? They can use various techniques. They can use jumping libraries that help join contigs into scaffolds. They can use long reads, sometimes in combination with short ones. But these techniques still quite expensive, and currently they have higher error rates compared to short reads. So maybe there is an alternative. How can we get a complete sequence without such advanced techniques? Of course, there are some methods. They're called reference-wisted assembly. And in the simplest case, we can map the contigs on a reference genome, and then join these contigs in the corresponding order. Very easy, but unfortunately, this method will not work if genomes have structural variations compared to each other. So we need some methods that are aware of possible genome rearrangements and that can handle them. Actually, there are some methods. In 2006, Gold and Blanchet proposed a procedure that orders contigs so as to minimize the number of structural variations between the contigs and the reference. And very recently, Kim and his colleagues proposed a tool called Ragout, which actually was the first attempt to use multiple references simultaneously. Actually, Ragout used one reference and multiple R-group genomes that also can help info order of the contigs. But actually, Ragout still heavily rely on this one reference. And both approaches sometimes make errors in presence of rearrangements. And maybe if we use multiple references, maybe we can reconstruct assembly more accurately. And we were working in this direction. And now I will tell you how to make a reference-wisted assembly with multiple references. I want to start from a very brief overview of Ragout. It is a tool, a program. It is written in Python, C++. The name is actually an acronym for Reference-Assisted Genome Ordering Utility. It takes as input multiple references in FASTA format, so we start from nucleotide level. It takes contigs or scaffolds, everything that genome assembler, short-read assembler output. And we also want a phylogenetic tree which includes all the references and target genome. And Ragout outputs scaffolds. Very simple. Now I will explain the algorithm. First, how do we represent a genome? We have to compare them somehow, but comparing nucleotide by nucleotide doesn't make sense. It is quite expensive, right? So instead, we want to extract some conserved regions. We call them Synteny blocks. And in this picture you can see decomposition of two genomes into Synteny blocks. But of course, we can decompose multiple genomes simultaneously using Siberian for bacterial genomes. And here I want to make an assumption. For this presentation, I want to say that each block is represented exactly once in each genome. The real algorithm that is described in paper, it works with real data, and it is perfectly analogous with except some pre-processing procedures. But for now, let's make such an assumption. And now our genome is represented as a permutation of Synteny blocks. So it is natural to represent it as to build a so-called genome graph. In this graph, we have directed black edges, which represent Synteny blocks, and we have undirected red edges that connects adjacent ends of adjacent Synteny blocks. And as you may notice, we consider circular genomes, as bacterial genomes are circular. Now we will join two such structures into one to compare the genome somehow. We have two genome graphs, P and Q, and first we will reorder black edges in P so they will be in the same order as in Q. And then we just add, we simply add green edges, and after we can remove black edges because they do not have any useful information for now. And we can build graph for multiple genomes in a similar way. And this graph has a useful property. Edges of each color define a perfect matching in this graph. So you see that green edges define perfect matching, blue edges define perfect matching, red edges define perfect matching. But as you may notice, this graph was constructed from complete genomes. What if the genome is fragmented? This will correspond to some missing adjacencies and missing edges in the graph. So you see that connection between CB and BD is broken, and we don't see corresponding edges in graph. So our task now can be reformulated. It can be first construct a breakpoint graph from all the references and the target, and then recover missing edges. And as we remember, these edges should define perfect matching, so all we have to do is recover it. But actually, there are a lot of variants of such matching. So how do we choose the correct one? To do this, let's first consider a genome rearrangement, and let's take a look on the graph. Here, BC is inverted to CB, and it corresponds to some operation. It corresponds to removal of two red edges and adding two new red edges. And as you see, if you define a state as an adjacent vertex and consider a node CT, then rearrangements will change states of this node. So state change from DT to AH. And now let's say that rearrangements are some operation that change the states of the nodes. Then first let's do a step back. Let's choose just an arbitrary perfect matching. Let's recover the graph with just an arbitrary perfect matching, and also consider a phylogenetic tree of corresponding genomes. Then let's choose a node in this graph and consider all the states that this node have in different genomes. So you see that BT is connected to CT in blue genome, and it means that it has corresponding state. And let's label the three nodes with these states. So we put CT in blue genome and so on. And now this tree represents evolution of state of this node in different genomes. Let's find the scenario how this evolution This is well-known Parsimony procedure, and what we want to do is to minimize the number of operations. And we can solve it with dynamic programming, and after, when it is solved, we can assign a cost, which will be just the sum over all the three branches in which states were changed, and we also incorporate the lengths of this branch. So now we can calculate state for a node, or cost for a node, sorry. Then we can cost the whole graph. Just it will be just the sum over all the nodes. And we want a perfect matching that minimizes this cost. How do we find this matching efficiently? Well, we can move from node weights to edge weight, where edge weight will be just the sum of two corresponding nodes. Then we can pre-calculate these weights for each possible edges in the graph, and then find a minimum weight perfect matching. And this matching will correspond to optimal order of contigs in our model, of course, optimal, and also to a certain order of the contigs. Well, this is great, but actually we have some problems to address. We still have some problems to address. One of them is a choice of Synteny block size, because big Synteny blocks are more reliable, they are easier to detect, but some contigs are too short to contain such blocks. To overcome this, we propose an Iterative Assembly procedure. What we do is we do multiple assemblies with different sizes of Synteny blocks, and then we merge them. The idea of merging is that we start from big contigs, like a skeleton, and then we fill it with small contigs. And the idea is that order of these contigs should be consistent. If the order is not consistent, then we keep big contigs because we trust them more. And this procedure makes us possible to make a reliable assembly, but incorporate as much contigs as we can. Also, there are still some very, very small contigs and also repetitive contigs left. To incorporate them, we use assembly graph, which can be reconstructed by overlapping the contigs. And the genome traverse of this graph is some unknown path. But now we have an information. We know that one big yellow guy follows another big yellow guy. And if they are connected with a unique path in the graph, we can insert contigs along this path into the assembly in corresponding positions. And it makes it possible to incorporate very, very short contigs and even double the number of contigs used. And actually, it is all. And now let's do some tests. Let's see how Ragout performs. First, I want to make a very easy test where we have only one reference. And it makes it possible to compare Ragout with other tools that use only one reference. There are MOV Contig Mover and OSLay. And these genomes do not have any structural variations between each other. And we measure number of scaffolds. In ideal case, number of scaffolds equals number of chromosomes, but as algorithms are imperfect, then sometimes we get more scaffolds. We also measure number of contigs and their coverage, bigger is better, and number of misordered contigs, number of errors. And as we can see, Ragout is not worse, and even better in terms of number of contigs incorporated. So we can conclude that Ragout passes a very basic test and can be used in usual assembly with one reference. Then let's move to more problematic case. We have 4 Helicobacter. Thank you. We have 4 Helicobacter pylori references, and each of these references has rearrangements compared to the target genome. And what we want to show is we want to show how number of references affect the results. So you can see that as number of references increases, number of errors decreases, and finally, when we have 4 references, we are able to get a perfect assembly without errors. And we also compared Ragout with Raka, which is actually only one tool available that can handle multiple references. And as you see, Raka is something worse in number of contigs, and it also has more scaffolds. Then, quite recently, in Nature Biotechnology paper, Ali Bashir and his colleagues proposed a method for automatic finishing of bacterial genomes with the hybrid assembly. In this study, they used very, very short Illumina reads, also 454 reads, and PacBio reads. And they managed to get a complete genome, complete assembly. And the question is, can we replace, in this study, can we replace long reads with Ragu? We took 3 references, and with 3 references, we get 2 complete scaffolds that corresponds to 2 chromosomes of Halira. And also, as you see, as the number of references increases, the number of misordering contigs decreases. And in this case, Raka is, also has no errors, but has lower coverage. So, as you see, using only short reads data, very, very short reads, and also they are non-paired, we can get nearly complete scaffolds using only short read data. All right. So now, very briefly, what we are going to do next. We are going to move Ragu into mammalian scale genomes, and we start from Drosophila. We have some results here. And next, we are going to move to multiple mouse species, mouse lines. And we also want to improve a procedure that captures rearrangements. And we are going to use assembly graph for this task. And we also are going to improve the usability of our tool and integrate it with Illumina-based space. That's all. I want to thank all of these people, especially my advisor, Sean Pham. And travel funding was provided by Akame Technologies. And here you can see a website where you can download and try Ragu. All right, thank you for your attention, and I will be glad to answer your questions.
Well, just a brief question. Wonderful work. How does it scale in terms of memory and time as you increase the genome sizes?
Well, actually, the most time-consuming part is Synteny block decomposition. For mammalian scale genomes, we use a multiple genome alignment. We use progressive cactus, and then we also have an algorithm that recovers Synteny blocks for multiple sequence alignment. For instance, if we have 5 Drosophila genomes, we can align them in 5 hours. And actually, Ragu part, breakpoint graph analysis part is very fast. It's like just a few seconds. And so, yes, the algorithm of analysis breakpoint graph itself is quite fast.
Thanks.
An impressive technical feat, and I have a kind of a biology question. So my understanding is this is really just about arrangement of your contigs. It's, it's a scaffolder.
Yep.
But so it's interesting to think about looking at the other, the sort of evolutionary inference of what the scaffolding might be. But I'm not actually seeing any evidence from the data to support the work that's being done here. These are different samples. There could be a major rearrangement, you don't know. So is your intent to use this program to say, well, this is a likely scaffolding, or is there some data there that I'm missing? Because I'm not seeing that there's actual evidence for that. Because it's from a different sample, it's from a different, it might be a not as closely related species.
You mean that we can trust the phylogenetic tree?
Yeah, I mean, I think that phylogenetic tree talks about different sampling events and possibly different species, but you're trying to do the assembly of, one particular sample. And I'm a little confused there because I'm not seeing that there's evidence in the data that support a particular rearrangement. So I was curious, are you presenting the output of this tool as this is the most likely, or this is the correct scaffolding for the contigs that we have, or is it just as this might be a useful way for doing comparisons later?
Well, currently we do not provide the likelihoods of a particular connections between the Synteny blocks, between the contigs. So we just construct the scaffolds output the scaffolds. But it actually is a good idea to measure the likelihoods of such connections. We didn't think about this yet.
Okay, I'd enjoy hearing more about what the particular goal was other than a scaffolding.
Thank you.